we'll start off with a little bit of an introduction to exactly what the GSC is, how we function, and how we operate. So the Genome Sciences is actually a department of the BC Cancer Agency, which is a branch of the Provincial Health Services Authority in British Columbia. And that's a little confusing to people at first who don't understand the structure of groups. We also have a lot of scientists here that hold dual appointments in various other universities, and a lot of those are with Simon Fraser University and University of British Columbia. So quite often we're dealing very closely with those two institutions, especially UBC, as a lot of our grants flow through UBC. Just to give some background information about the GSC, we've been around since about 1999. We have a College of American Pathologists certification that started in 2011. That's for some of our wet lab space. We became a Compute Canada node in 2013 and have a little bit over 300 staff here. And that's a combination of people who work on the grant, scientists, a lot of bioinformaticians. We also have a large contingent of trainees, postdocs, master's students, co-op students, all that operate and work here. We have a fairly large cluster on site, about 18,000 cores and about 12,000 GPU cores. I'll talk a little bit more about those two in a bit, and quite a bit of storage. Genomic data, for anybody who is familiar with it, is large. It's not necessarily big data, as our IT director often likes to say. It's just lots of data. So we have lots of files, and that constitutes about 12 petabytes of storage. A spinning disk, about the same amount of disk backup in tape library form. So I wanted to just explain a little bit. We've heard a lot from people wondering about, if they have bioinformatics sort of questions, how can the GSC help with that? We have, as I mentioned, over 100 bioinformatics personnel here that have a whole variety of different specialties and experience. So some of those people are very experienced with visualization. Some of them are more into the programming. Some of them are more into the analysis. It really, really varies. And because of that, depending on the questions somebody might have where they need to get a little bit more information, it really would depend on what the question is as to who would be best to answer it. So rather than having a single person that we just say, okay, always ask this individual, we've set up so that you can access that kind of support resource by going through the westgrid.ca support email address. And that will eventually route through to really the best person to answer that question. And it can be all manner of different bioinformatics sorts of questions. And they're people who are generally very interested in helping. I wanted to touch on bioinformatics resource here that is fairly new called ORCA. ORCA is something that was developed a little while ago to allow people who have a need to access bioinformatics tools direct access to a containerized environment that contains a whole series of pre-installed tools that are verified to be operating. And they can do that in a secure way and a fairly easy access methodology. There's not a whole bunch of forms to sign or details to go into. You can access this fairly readily. We've got about 200 tools in it now. We're constantly enhancing this and making it better. And it's got some good, strong hardware that's backing it up. So there's actually some resources there. There's a website specifically for it, and the link is on that there. And just to go into it a little bit more, it's basically, as I mentioned, a container. You SSH into it, you'd load your data into it, and then you're ready to go. And you can access any one of the tools that's in that container. It comes with about 64 gigs of RAM for each individual, and we start off with 50 gigs of storage, and that's backed up storage. The key thing with that one is that's just our default to start with 50. It's a fairly usable amount of space for most bioinformatics pipelines. But if you need more, it's something we could definitely discuss, and it would just be a matter of talking about what the needs actually are. But we found that 50 was a good starting point, a good way to get going. It's a private container in a shared environment, so that 64 gigs of RAM is a cap on it. But if a whole bunch of people are using it at once, it will slow down the system a little bit because it is shared across a couple of different nodes. And some of the nice things it'll do, though, is we can allow it to run quite long duration jobs. If you have a job that needs to run for a few days, that's fine. You can log in, start your job, leave, when you come back, you'll go right back into the same container, and you can check on the status of that job, see how it's running. And when it's done, you can SSH your data back out. We also maintain persistent software versions. So even if we're updating the software versions that are in the container system, we're going to provide consistent access to what was there before. So you won't hit any batch effects if you're changing software versions or anything like that. What it's not is an HPC cluster. So it is backed off an individual node. It operates like an individual node. So if you need something with a lot more oomph behind it, something that actually has a whole cluster backing it, then this isn't the right tool for that. It's a very good tool for relatively small pipelines or experimenting with pipelines or experimenting with smaller data sets. If you need a whole HPC cluster backing, that's not what Orca is designed to do. And it's also not GUI-based. So you do need to have some experience with the command line in order to use it. As I said, the tools are already there. They're installed. They're ready to go. And that's where the real benefit lies. But it's not something that's going to guide you through with some sort of a GUI interface. Another one of the things that is available at the GSC is co-location for various systems. And we're doing this with a bunch of different people now. We try to really hone this down into systems that are closely related to what we're already doing. So somebody who's doing bioinformatics and has a system and they need to put it in place where they're dealing with clinical data, personal health information. They need a systems team that's experienced with what I'm calling genome scale data storage. So this is where you're getting individual files that easily can exceed, multiple gigabytes or hundreds of gigabytes in size. This is a little bit different than Orca. We do this through a collaborative SOW. And it would be something that our team and whoever has this need would discuss and figure out exactly how best to make it work. As I say, we're doing this with a bunch of researchers now. It's just a way to provide them access when they have a smaller cluster and need a place to put it and maybe want to leverage some of the existing other infrastructure that's already in place at the GSC, like the backup and the networking and everything that's already here. Another thing I thought I would touch on, and this is coming soon, it's already here, but we're just finishing hooking it up to a network in a way that everybody can access it, is a fairly powerful box from NVIDIA called a dev box. It's basically got 4 Titan GPUs in it and 12 gigs of RAM in each one. And it's a very, very beefy GPU box that you can use for experimentation. So if you want to play around with accelerators and see how that can affect various pipelines and software, that's what this box is exactly designed to do from NVIDIA. They've got some additional ones they're planning to come out with and sell, but if you wanted to see how the GPUs are going to affect software applications that you might use on a regular basis, this is something we'll be making available to people to come in and play with and see how it affects what their pipeline might do. So I wanted to touch just briefly on some of the other things that the GSC is doing, and from a scientific point of view. One of the key things that happens here quite a bit is a lot of software development, again, in the bioinformatics area. So there's some, if you're into bioinformatics at all, you'll probably recognize some of these, Abyss, Circo, Spark. There's a whole list of these on our website in the software section, and constantly being enhanced and improved to provide better functionality, and they're all available to academics across the country and across the world, really. We do a lot of collaboration internationally. One of the ones that's also on this slide is a collaboration we had with Intel, and that was really to further parallelize some of these software applications and find great performance enhancement through vectorization of the code and making things work a lot faster with the newer multi-core processors, things like the Xeon Phi, the upcoming Intel architectures that are almost fully out into market, and really leveraging those multi-core architectures to provide even faster bioinformatic analysis. We also have a collaboration with IBM Watson to look at genomic analysis. And there's a whole series of other international, national, and regional collaborations going on with those bioinformatics specialists I mentioned before, and centering around software. And there's a great deal of resource here, and I'm going to give a little plug, especially around the Circos and the visualization side, that sometime in the new year we'll be having one of those specialists give a talk on bioinformatic visualization. And he's actually the developer of Circos. amongst other things, and is very knowledgeable about that. And he's one of the people that you would be able to access if you had bioinformatic visualization questions. So that's just another area where we like to be able to help the community.
I have a question.
Sure.
I just went to your website and I couldn't find the program Trinity. Is there a reason why the program is not here?
I'm not familiar with that specific one. I would have to go and look, but I can find out for you and get back to you.
Okay, thank you.
Sure. So I'm going to hand it over now to Diane Miller, who is a colleague of mine here, and she's going to talk more about the actual sequencing side of the GSC and the services we provide in that area.
Hi, everyone. Scott said, I am Diane Miller. I am a projects team leader here, and I work with collaborators who wish to access our sequencing services generally. To give you a bit of an overview of our pipelines, we have a series of pipelines at the GSC, starting with our biospecimen core, who receive samples, our library core, who will build libraries or sequencing libraries from the samples, sequencing core, who will sequence the samples, informatics core, and a proteomics core. All of these cores have input from the IT systems and LIMS group, management and admin, quality assurance, and our technical development group. The services we offer, if you're interested in sequencing, there's some examples here. So next generation Illumina sequencing is the main service we provide. For example, genome, transcriptome, microRNA, epigenetic modifications, and some special capture work. We still offer high-throughput capillary sequencing for people who are looking for ESTs or back-end sequencing. And we have a service for T-cell and B-cell receptor profiling. We have bioinformatics services. So this would be for people who would like some assistance in analyzing the data that is generated through our sequencing. We have both standard modules and custom analysis. So we have standard genome annotation or high-throughput PCR prediction modules, but if you need more customized analysis to help a particular project answer a very specific question, we definitely can work very closely with a project to provide that. We have a proteomics service for protein-protein interaction mapping, phosphopeptide, phosphoprotein analysis, protein isoform detection and quantification, and we also have an in silico drug design group. For those who are interested in the actual technology, so the nuts and bolts, for our sequencing, our current capacity is 12,000 30x human genomes per year. We have generated more than 1.2 petabases of sequencing data so far. Our lab currently holds 5 HiSeq X instruments, 4 HiSeq 2500s, all of which have been updated to the 1 terabase configuration. 3 MiSeqs and 2 NextSeq 500s, and our 2 capillary ABI 3730s. Proteomics has a Thermo Fisher Orbitrap Fusion MS, an AB SciEx 56 Plus triple TOF, and a couple of other AB instruments. So to give an example, one of our main goals here at the GSC is to share access to technology pipelines and expertise to enable science. And as an example, we are a Genome Canada platform and are involved in Genome Canada and Genome BC projects. Currently, we are also involved in CFI and BC KDF projects, and as you all know, a Compute Canada node. We're a TCGA partner. We are involved in reference epigenome mapping centers, and we're also partners with Stand Up to Cancer grants. This is just an example of the current grants that we are involved with, and this will of course change regularly. So how does the process work? How does somebody come to us if they have an idea? It's really very straightforward. If somebody has an idea for a scientific project, and they would contact us either by email or on the phone, and give us a bit of an idea of what they're looking for. So what do you need? An idea if you have a budget, so you might have a strict budget, and you may have some ideas you may need to have some data in a certain amount of time. And we would work with our internal scientific team and have technical discussions as required until we develop what we call a statement of work. And this is just a document that defines the scope of work of what will be submitted, but also what will be delivered all the costs associated, includes our terms and conditions. We need to have it signed, and our website has a lot of information of the terms and conditions. So there's generally an iterative process. So somebody might come with an idea, we might provide some costs, and then we may refine it based on the needs of the project. Finally, we have completed activities where we would provide the sequencing data, for example, or obviously if you're asking for proteomics, you get the proteomics data. And finally, we would follow up with invoice, which is basically cost recovery. So we just recover the costs of the work that has been performed. To give you some idea, we collaborate with people all over the world. Here is a map of some of our collaborative centers. We obviously are mostly collaborative within North America, but we do work with any country. I've never turned anybody away, so it's kind of fun. And how do you access the resources? That's me. You would start here at our website and look at the services. There's an email address provided and a phone number. And if people want more information, they are welcome to contact me at any time. And if there are any questions, I am specifically equipped to answer the sequencing questions. And so perhaps we can let me know when we need to move back to Scott for the technical IT questions.
Great, so we'll open the floor to questions. If anyone who's connected by video would like to ask a question, please feel free to unmute yourself by clicking on the microphone icon, and feel free to speak. And currently, I don't have any questions received by email, but I will let you know if they come in. So does anyone on video have any questions? Okay. No further questions have come in via email. I suppose if that's it, then we've got your contact information. Scott and/or Diane, if you have any last words that you would like to share.
I can share one more last little tidbit, and that is we do have another plug. We do have another webinar coming up for anybody who's more interested in the ORCA platform. One of our IT specialists here is going to be giving a 1-hour training in how to use ORCA, how to get into it, basically how it works, some hands-on examples of how it's going to work for you. So anybody who's interested in that sort of a platform should have a look at the WestGrid site for the next upcoming webinar where he'll be going through that process.
Great. Okay, thanks very much to Scott and Diane for connecting and sharing that information today. As mentioned, we have recorded this session, so we'll be posting this to the WestGrid website in the next day or so, and I'll email out a link to that recorded video and a copy of these slides to everyone who's RSVP'd today, so that you've got that for reference.
Yeah?
Yep.
I have a general question. If you were to compare ORCA with the current resources that WestGrid has, like HangaBee, that is an ideal cluster for programs like Trinity that are kind of big huggers that run well between cores but not between nodes. How would you compare ORCA with those clusters?
There's little comparison, really, with a cluster, because ORCA is just a single node environment. The main difference is something that is an actual cluster-based is designed to handle big workloads, as you mentioned, all across multiple nodes, multiple cores. ORCA's main goal is to handle small workloads where people don't want to spend the time messing around with installing various software applications. So it contains all the software that we could bring into it from homebrew science and Linux brew related to bioinformatics, and it's pre-installed. So that's the big difference. It's just an environment where everything that you might want is hopefully already there. And if something isn't there, then it's definitely something we could explore trying to add into it. But the goal with it is to really provide a ready-to-go environment rather than to provide a whole cluster. So if you knew you had some cluster work to do, this could be an area where you could try out some things first, get your pipeline polished up, make sure everything's working the way you want to do it before you start using up whole cluster wall clock hours.
Right. So two follow-ups to that. One is that the kind of programs that I use don't really work between nodes, and that is one of the reasons why I need big clusters like HungaBee that gives me a lot of power within one node. So that would not be unusual then. But the follow-up to the installation programs is that Westcliff also does a lot of the installation for the programs for us. But that itself can be a little troublesome when you just want to test a program. And like in my computer, I can install it through my path and whatnot, but in Westcliff, I have to request it from the cluster itself, and that puts a limit into how many programs I can test and, to what level itself.
Right. Yeah, and that's sort of where Orca's trying to fill in a gap, is that you can go in there and you can install stuff and test stuff in it. It comes with all that stuff pre-installed. It doesn't prevent you from trying to install more.
Oh, that's great. So you can install your own programs then independently from having to request it from you guys.
Yes, absolutely. It's basically a private environment that is there for you to play with and use.
Great. That's a big difference. Thank you.
Okay, sure.
Great. Any other questions from anyone? Okay. We have no questions through email from the web stream viewers, so we'll wrap it up there. I'd like to thank both Scott and Diane for presenting today, and look forward to the follow-up session. I believe it's December 1st on the Introduction to Orca to learn more about that. And so that would be a good one to answer even more questions about how that can work as a private environment. Thanks, everyone, and we will end the call.
Thank you.
Thanks.